to start obstetricians and gynecology quick revision series. So first let us start with gametogenesis. So in the name itself here you can see gametogenesis means giving rise to gametes. So we have female gametes, male gametes, right? So first let us see the introduction of this gametogenesis. Actually this germ cell, there is called a germ cell, and that germ cell mainly originates from epiblast. Okay, so go back to embryology, you have epiblast, right? So actually from that epiblast the germ cells come. Okay, and later what happens is from here the germ cell go to the yolk sac. So when it will reach the yolk sac, it will reach by third week. So by third week the germ cell will go and reach the yolk sac. Okay, so from the yolk sac the germ cell will migrate to the gonadal ridge. Okay, so it will go to the gonadal ridge. Of course, in the case of male it will go to male gonadal ridge, in the case of female it will go to female gonadal ridge. Okay, so this will take place by 6th week. So this is also important, this time period. Okay, so first you have a germ cell, it is formed by epiblast. So from the epiblast the germ cell is transferring to the yolk sac by third week, and from the yolk sac it will go to the gonadal ridge by 6th week. So now it has come to the gonadal ridge, right? So now one more thing you need to remember, that this germ cell is bipotential. It's bipotential till 6th week. Means it can either be a male or female. It may either convert into male or female. So who is going to decide whether the germ cell now which is formed as a male or female? The main region is called SRY region. So what is this? It is a sex determining region. Okay, so the sex determining region is present on Y chromosome, hence the name SRY. Okay, sex related region of Y chromosome. That too, on what part of Y chromosome it is present? It is present on short arm of Y chromosome. So how to remember this? Actually I remember like this. I write the Y like this because here this is a short arm, like that I'll remember. Okay, so mainly the germ cell which has come, it is determined either male or female by means of this SRY region. Okay, that is present on Y chromosome. So now we have decided it's either male or female. Okay, for example, if it is a male, then it is called Spermatogonia. Okay, Spermatogonia. And if it is a female, then you will say Oogonia. Okay, so of course the chromosome number will be 46 in the case of Spermatogonia, XY. Okay, and in the case of female Oogonia, 46XX. Okay, or also you can write it as 44 plus XY, here also 44 plus XX. Okay, so like this will determine either male or female. So after this the main process starts, that is gametogenesis. So these cells now will undergo the differentiation process. So how the differentiation process may be? Either mitosis or meiosis. And you know that mitosis is where the chromosome number remains same, whereas meiosis is divided into meiosis 1 and meiosis 2. Okay, in meiosis 1 it's a reductional division, that is the chromosome will reduce. So I would like this. Okay, whereas in meiosis 2, same like mitosis, no chromosomal change, chromosome remains like that. Okay, so this is a introduction of gametogenesis. So I hope you have understood what is going on here. So now let us start the main part, what happens in male, like what is going on. It is nothing but the process is called as spermatogenesis. The name itself you can see genesis, that is a sperm is going to born. Now how it is the process, what is going on, let us see. So spermatogenesis, as we said, first will determine either male or female. So this spermatogenesis process is going to start by means of our spermatogonia. This spermatogonia will convert into sperm after this process. Okay, so it will going to convert into sperm after this process, and the spermatid are going to convert into again the same sperm. Okay, So what are these processes called? This process of spermatogonia to sperm is called spermatogenesis. This process of spermatid to sperm is called spermiogenesis. Mio. Okay, here mato, here mio. So don't confuse between them. So now we are clear with this. Actually, this process of spermatogenesis in the male starts exactly at puberty. Okay, this process starts at puberty. And remember that one primary spermatocyte can give rise to like about 4 sperms. Okay, so one spermatocyte, primary spermatocyte, have a chance, have a capacity to produce 4 sperms. Okay, so this is important. Now let us start the process of spermatogenesis. First I said there is spermatogonia. This spermatogonia, first it will undergo the process of mitosis. By mitosis it will form a primary spermatocyte. This primary spermatocyte will undergo the process of meiosis 1 and it will form 2 secondary spermatocytes, and these 2 secondary spermatocytes will in turn undergo meiosis 2. Okay, by meiosis 2 each of it will form 2 spermatids. That is, in total you will get 4 spermatids. These are called spermatids. So this spermatid will be having the chromosome as X. They'll be having Y. Okay, so now the spermatid they'll undergo a transformation process, or there is no meiosis or mitosis in this process. As I already said, this is called spermyogenesis. That is, a spermatid is now converting into 4 sperms like this. Okay, 1 2 3 4 sperms. Actually this is a tail. Okay, so finally this is how the spermatogenesis process is going on. So actually, as I said, primary spermatocyte has a capacity to form 4 sperms, and this one more thing is the one spermatogonia. This spermatogonia have a capacity to form 16 primary spermatocytes. Then if 16 primary spermatocytes are forming and each of it is having a capacity to form 4 sperms, then you can count 16 into 4 will be 64. So like this, 64 sperms can be formed by one primary spermatocyte. Okay, and one more thing is, daily on average there is formation of about 100 million sperms per day. 100 million sperms per day. And this process, this overall spermatogenesis process, takes the time of like about 72 to 74 hours. So it takes this much time to undergo this process. Okay, so these are few important points. Now let us understand spermiogenesis process. Okay, so now we are going to discuss about spermiogenesis. So as I said, this is a process, nothing but the spermatids which are formed are going to convert into sperms, right? So in this process, remember, it is just a transformation process. There is no mitosis, there are no meiosis in this process. So now let us draw the sperm like this. So actually, if you see this structure, this is the acrosome, this is the head, this is the middle piece, this is a tail. Okay, so actually the spermatid will lead to form the sperm structures here. The Golgi apparatus of the spermatid forms the acrosome. Okay, the nucleus of the spermatid forms the head. The mitochondria of the spermatid forms the middle piece, and the tail is formed by the centrioles. Okay, remember that as the mitochondria is present in middle piece, it is a powerhouse for the sperm. So the middle piece is a powerhouse for the sperm. Okay, so if you see, I mentioned about all the cell organelles but not about the endoplasmic reticulum, because the sperm doesn't have the endoplasmic reticulum, especially the rough endoplasmic reticulum is absent in the case of this sperm structure. Okay, and these things are important. next coming to the tail. If you see in this tail, as I said, there is a centriole and it is having a 9+2 arrangement. Okay, there is 9+2 arrangement in the tail. Now coming to the length of the sperm, the length is like about 55 to 60 microns. This is important. 55 to 60 microns is the length of the sperm. And next, its lifespan. Its lifespan is like about 48 to 73 hours. This is very important. Lifespan is 48 to 73 hours. Okay, so now coming to the location of this spermatogenesis process, the location is at seminiferous tubules. Okay, so in seminiferous tubules you have this spermatogenesis process, and actually where the sperm gets its motility, the motility is at caudal portion of epididymis. Okay, caudal portion of epididymis. And I already said this process undergoes at the age of puberty. Okay, motility at epidermis. And for the motility there is a protein called Caspar, and there is an ion which is required for motility is calcium ion. Okay, so these are all like important points which you need to remember. Now coming to the so what is the time period where the sperm it is staying in female genital tract? In female genital tract it stays up to the 12 hours of time. Okay, and to reach the ampulla, the sperm takes just 30 minutes to reach the ampulla of female genital tract. So now coming to the hormonal support. So what is the hormonal support for the sperm? It is mainly from the hypothalamus. From the hypothalamus, gonadotropin releasing hormone will be released. It will go to the anterior pituitary. From the anterior pituitary, it will go to the it will lead to the release of two hormones, that is FSH and LH. FSH mainly act on the Sertoli cells. LH will act on Leydig cells. So how to remember this? LH on Leydig, FSH on Sertoli. Okay. So FSH acting on Sertoli will produce inhibin B. This inhibin B, and also there is ABP, that is androgen binding protein. So this inhibin B have a negative feedback effect on the FSH. Coming to LH, as I said, it will produce Leydig cell. This Leydig cell will in turn form the testosterone. This testosterone is in turn having a negative feedback on luteinizing hormone. Actually, testosterone is not formed only by LH effect, even FSH and LH, all are the factors. So finally, by this testosterone only our spermatogenesis process is taking place. Okay, so testosterone is important. So now coming to another feature, that is the blood testis barrier. The blood testis barrier is present especially for our Sertoli cells. Okay, Leydig cells doesn't have any blood testis barriers. And next coming to testosterone. Actually, testosterone is produced in 8th week of intrauterine life itself, whereas it will get into peak by 15th week. Finally, coming to the stimulus, like who is saying for the testosterone to come out? HCG. So HCG stimulates the production of testosterone. HCG. Okay. And finally, the testosterone will convert itself into a powerful form by 5-alpha reductase enzyme acting on it and form a powerful potent testosterone, that is a dihydrotestosterone. Okay, so this is all about male process. In the next video we'll discuss about female process, that is oogenesis. Okay.